Hey, I'm John Wherry. I'm at the University of Pennsylvania. I'm the chair of the Department of Systems Pharmacology and Translational Therapeutics and the director of the Institute for Immunology. I'm also the associate director of the Cancer Research Institute Scientific Advisory Council. There are really two things in this morning's cell therapy session I thought were really exciting. The first, that's been developing for some time, is the development of TCR therapies targeting driver mutations. So KRAS, G12D, or G12C, specific TCRs are starting to show some promise in the clinic, and we saw an interesting new trial using Sleeping Beauty to transduce T cells that showed some really promising new data. The second set of data was actually some extremely promising CAR T cell therapy data in solid tumors from Ugur Sahin and BioNTech. Both good CAR T cells, but also boosting those CAR T cells after adoptive transfer with vaccination seemed to give really promising responses in solid cancer. Outside of the central nervous system, this is really the first good solid tumor CAR T cell data that I've seen. Yeah, I think one other aspect of the cellular therapy session that was interesting was the importance of identifying new targets. We heard a couple different talks that used both looking at T cell responses in non-cancer patients to identify T cell receptors that can target neoepitopes or cancer antigens, thinking that the immune response in cancer patients may be depleted of those cells and we need to find them somewhere else. We also heard a couple different talks that highlighted the need to identify new antigens, new targets for CAR T cells, new targets for TCRs, and we need to expand our repertoire of things that we can target with either CARs or TCRs. One of those talks looked at the importance of glycosylation or sugar modification of proteins and the ability to recognize antigens that really are sort of the combination of the protein backbone and the truncated sugar that's attached to those proteins. And they open up a whole new set of targets that we can use for making new CAR T cells. I think we're just scratching the surface of understanding the overall immune ecosystem and how it impacts cancer. What we know from autoimmunity, if you look at twins, is that only about 30% of the penetrance of autoimmunity is genetic, even though we think about autoimmunity as a genetic predisposition or a genetic disease. What that tells us is 70% of the influence for a disease like autoimmunity is environmental. Probably immune history. It's the way your immune system has evolved over your lifetime and incorporated previous immune experiences into the ability to mount future immune responses. We have to start integrating this idea into cancer immunotherapy as well. We heard great talks about the overall immune ecosystem, the role of systemic immunity in local tumor responses, and on the role of the microbiome and intestinal health and nutrition and how that impacts the ability to mount immune responses against cancer and respond to immunotherapies. So, thinking about the immune ecosystem in a patient, the immune system monitors everything in all our tissues all the time, and then those cells mix in our blood. And so the challenge of measuring everything over time in all patients, integrating it with clinical data, integrating it with what the patient eats, how they sleep, really creates a huge challenge, not just with the scale of the data, which is massive and something that we all struggle with even now, but also how do you integrate this? How do you make different data types play together? It's pretty easy when you think about what I shouldn't say easy, but the genomic revolution over the past 10 years has transformed the way we think about science. But there's been one big advantage. That is, for the genomic revolution, when you sequence DNA or RNA, it's still an A, C, T, or G. So putting that data into context and actually integrating it, making it compatible across different studies, has a lot of advantages to data sets that actually have imaging data from radiology, biometric data on how often you sleep, flow cytometry data on the immune cells, all sorts of other features that we have to actually think about how to integrate. So one of our big challenges in cancer immunotherapy is going to be wrestling with this data, not just the scale of the data, but how to actually integrate the data and turn information into knowledge and turn that knowledge into actionability. And so I think there are a couple steps that we're going to have to work very hard on over the next 5 to 10 years. Well, I think I have some optimism about cellular therapy. We've seen from Crystal Mackel's talk at the very beginning, to Ugur Sahin's talk today, to the idea that we're discovering new antigens and have TCRs against driver mutations. It feels like that field is just about ready for kind of a transformation in the solid tumor space. we're going to be using the same technology, so this isn't quite a step change, but we're finally solving some of the engineering type challenges to make a big difference in solid cancer patients, and there's plenty of work coming on that. So I'm optimistic that we're going to see some positive impact. I'm also encouraged and excited by the fact that there are people appreciating and trying to understand the immune response in cancer patients beyond the tumor. And I think this will help us put things in context and understand how to integrate immunotherapy with the other things that happen in these patients. It's fascinating to me that we can give immunotherapy and chemotherapy at the same time. Chemotherapy is designed to kill cells that are dividing, and immunotherapy is designed to cause immune cells to divide. How we can put these two things together and actually get effective clinical responses is mind-boggling to me. We have to think about how to use the patient's immune information more broadly to inform how to combine immunotherapy with other kinds of therapy and boost the overall immune architecture of the patient to boost immunotherapy.